Today, we'll begin talking about the basics of genetics for understanding human evolution. Throughout the years, people have asked, where do we come from? In the past, we also wondered whether life forms had changed. Hundreds of years ago, people believed in creationism, the idea that the world and everything in it was created divinely all at once, and life forms are immutable. That is, they don't change. Linnaeus believed this. But by the end of the 1700s, most people had accepted that life forms had changed. But the question that we'll talk about today is by what mechanisms? Lamarck proposed that they change through inheritance of acquired characteristics. He proposed that during the lifetime of an individual, changes occur that are passed on to an offspring. Although this has been discounted, now we actually accept it in a tiny bit in a what we call epigenetics. Instead, what we will accept in this class is the theory of natural selection, which states several things. 1, organisms are adapted to their environment. 2, variation already exists among a population. And 3, population undergoes adaptive change when the environment changes. How? Those members of the population best adapted, that is, to the new environmental conditions, will survive. And those who survive pass on their variation to the next generation. There is no direction to evolution. Populations change as a result of natural selection through survival of the fittest. For example, let's say that in the environment of today, the variation within the population of humans looks like this. But the environment changes, and suddenly humans to the left of the dotted line can't survive, and only those humans to the right can survive. Then in the next generation, the distribution of variation in the population will look different. The theory of natural selection was formulated by two men, Alfred Russel Wallace and Charles Darwin. Although Wallace published first, Darwin had thought of natural selection 20 years before, but was afraid to publish. But when his book came out in 1859, there was an immediate acclaim. The entire first edition was sold out the first day that it appeared. Now, Darwin and Wallace didn't know the mechanisms by which variation was present in populations. It was Gregor Mendel in 1866 who sped us on the way to understanding the genetics of why variation exists. But even though he found this out in 1866, his work was not rediscovered until 1900. So the key difference in the two theories between Darwin and Lamarck, Lamarck said that inheritance of acquired characteristics, meaning variation arises when it's needed, versus the theory of natural selection says that variation already exists and nature does not select against those variations that are best suited to the current environment. So, taking an overview from the beginning week of classes, there has been change through time in organisms and in geology. And this change has not been directional. And biological change can be explained through the theory of natural selection. There are 5 main points to remember in the theory of natural selection: that organisms are adapted to their environment, that variation already exists within the population, and a population undergoes adaptive change when the environment changes. Why evolve when the environment changes? Well, those best adapted to the new environment survive. And those who survive pass on their variation to their offspring. A classic example of showing how population changes when the environment changes is the peppered moth. Prior to the Industrial Revolution in Europe, this moth had 2 different color forms, a black form, which was somewhat rare, and a lighter form. The black form was easily seen on tree bark and picked off by birds, and therefore fewer of them were passing on genes to the next generation. But during the Industrial Revolution, coal dust coated tree barks, and suddenly the white form became less adaptive and the black form more adaptive, and therefore more of the black forms lived and became a larger part of the population. So today we'll talk about the last 2 points that I mentioned before. What is the source of variation, and how is that variation passed on to offspring? Now let me point out right now that in talking about evolution, we must talk ultimately about populations, not individuals. But we need to start today by talking about how sexual reproduction creates individuals with variation. Mendel discovered that inheritance of characteristics is particulate rather than by blending, that traits are controlled separately by individual particles called genes. In 1953, Watson and Crick discovered that genes are portions of DNA molecules. So a gene is a chemical code or blueprint for producing a protein, some of which make up the basic structure of a cell in the form of enzymes. Each genetic code is made up of a sequence of bases that are part of a chemical strand called DNA. There are only 4 bases called by their first initials, A, T, C, and G. Bases are arranged in groups of 3, called a codon. Each codon makes an amino acid, and a chain of amino acids supplies the blueprint for a protein. Humans can produce at least 100,000 different proteins. Here it is illustrated a different way. 3 bases make a codon. 1 codon makes an amino acid, and a chain of codons or amino acids supplies the blueprint for making a protein. Proteins act alone or in complexes to perform many cellular functions. A gene is a segment of DNA that dictates the sequence of amino acids to form a particular protein. So each gene is located in a specific location or locus on the DNA strand. Here, for example, you see the locus for eye color. And here you see the locus for hair color. They each have a specific location or address on the DNA strand. Note that locus is another Latin-derived word, so the singular is locus, but plural you would say loci. To recap, a cell has a nucleus. Within the nucleus is DNA. DNA is made up of 4 bases, A, T, G, and C. And a series of 3 bases makes up a codon. Each codon makes an amino acid. A group of codons makes a gene, which makes a protein. And a gene has a particular locus or location on the DNA strand, or chromosome, and directs the development of certain traits. So a locus is an alternative word for gene. Most of the time, DNA exists uncoiled in the cell's nucleus. However, during cell division, DNA becomes tightly coiled into chromosomes. Each species has a characteristic number of chromosomes. Humans have 46 chromosomes in 23 pairs. Being in pairs is called diploid. Again, another Latin-based word, di, meaning 2, ploid, meaning number. During meiosis, chromosome pairs split, resulting in an offspring receiving half their genetic material from each parent, or the haploid number of chromosomes. Haploid refers to being single. The number of chromosomes you have is not related to how complex you are. Humans have 46 chromosomes. Goldfish have 94. Brine shrimp have 320. And a cabbage has 18. Genome refers to the total genetic endowment of an organism, such as a human. By 2003, the entire human genome had been studied for 2 humans. What was discovered was that 98% of the genome is not composed of genes that code for proteins. It's what is called non-coding DNA. And we are still learning the functions of this non-coding DNA and still trying to figure out the loci of all the genes. So like chromosomes, genetic loci also come in pairs. As we mentioned, a gene has a particular locus on a chromosome. So variations of a particular gene at a specific locus are called alleles. So each allele in a pair may be the same, such as you see here, or as you see here. You can see that these are called homozygous alleles, or they may differ from each other. They may be heterozygous. A heterozygous allele directs a cell to make a slightly different form of the same protein. So an allele results from a change in the codons. Where does a different allele come from? It comes from mutation, which is a random change in a gene. Different alleles can cause different expression of a trait, and we'll talk about that in a separate lecture. So here again, it shows a pair of chromosomes. And down here, you can see that these 2 alleles are the same. These 2 alleles are the same, but these 2 are different. These are heterozygous. In sexual reproduction, the organism produces special sex cells, or gametes. To do so, a cell passes through meiosis, which splits the chromosome pairs and therefore the allele pairs of the adult. Initially, in meiosis, the cells double their material, but then they split twice, ending up, instead of being diploid, but being haploid, with just a single set of chromosomes instead of a pair. So each sex cell or gamete, meaning each egg or sperm, has only half the genetic information of a regular cell. Now the alleles you inherit from your parents are passed down through these gametes. You will inherit only one of your allele pair from your mother and the other from your father. When both alleles in the pair are the same, they are called homozygous. Another Latin-based word, homo, the same. When the alleles in a pair are different, they are heterozygous. So remember, one allele in the pair came from your mother and one came from your father. The alleles that you possess define your genotype or hereditary makeup. But the way that they are expressed physically, what people can see on the outside, is called your phenotype, the sum of observable traits. So your phenotype does not always reflect your genotype. During meiosis, the first cell doubles and then splits twice. In the second step, the chromosomes split in half, with each daughter cell receiving a single-stranded chromosome. So the result is 4 daughter cells, or gametes, otherwise known as eggs or sperms, that each have 23 single-stranded, that is, haploid, chromosomes. And when they split regardless of whether that half of the chromosome had come originally from the father or the mother. That is, for you, from the grandfather or the grandmother. Mendel called this effect segregation. Once again on the right, here it's showing meiosis, the splitting into gametes or sex cells, the splitting into haploid chromosomes. Also during meiosis, sometimes homologous or partner chromosomes exchange information before they split in a process called crossing over. In crossing over, the 2 chromosomes in a pair exchange genetic information, here shown by different colors. So, whereas formerly perhaps this chromosome pair was all light blue, now it's switched some information, and the result is called recombination. So thus, you have new genetic combinations, but not new genes. Also, sometimes during meiosis, instead of sorting independently, regardless of who was the grandmother, who was the grandfather, where that allele originally came, sometimes genes on a chromosome may stick together, something that is called linked genes. To summarize, sexual reproduction is one source of the variation that is already present in a population. And the sources of individual variation that may arise during sexual reproduction include mutation, segregation or independent assortment, linked genes, and crossing over, resulting in recombination. So what is mutation? It's a random change in a gene, that is, in the codons that make up a gene. At the molecular level, this is the ultimate source of all variation. Now, unless the mutation occurs in the gamete, it will not be passed on to the next generation. So, for example, if you got a mutation in your little fingernail, that would not be passed on to your children. It's only a mutation that occurs in the gamete, that is, in the egg or in the sperm. Segregation, independent assortment refers to how chromosomes sort independently during the formation of the gametes. That is, they are distributed without regard to the origin, whether each side came from the grandmother or the grandfather. In humans, every time gametes are produced, 23 different chromosomes are sorted independently, which is 2 to the 23rd power, or over 8 million possible ways the chromosomes can combine to form a single gamete. However, sometimes instead of sorting independently, sometimes a segment of a chromosome will stay together, what is called linked genes. As we mentioned earlier, during meiosis, paired chromosomes may sometimes exchange material, that is, cross over, which results in recombination. So before they split into the gametes, they exchange material with the alleles on the other side, or the genes on the other side. So if 2 chromosomes contain different mutations on each side of the crossing over exchange, and they split during meiosis, the gametes they form could have a completely new recombination of alleles. So when one chromosome from the mother, which has over 8 million possibilities, combines with one chromosome from the father, which also has over 8 million possibilities, that is 2 to the 23rd by 2 to the 23rd, or about 70 trillion possible zygotes. In other words, you are 1 in 70 trillion possibilities at birth. So remember the theory of natural selection, which has 5 main points to remember. Organisms are adapted to their environment. Variation already exists within the population, and we've been talking now about why that variation already exists. The population undergoes adaptive change when the environment changes. Those best adapted to the new environment survive, and those who survive pass on their variation to their offspring. And one source of variation is sexual reproduction. So the sources of variation that may arise during sexual reproduction include mutation, segregation, independent assortment, linked genes, and crossing over, resulting in recombination. So these are sources of individual variation that are present in a population because of sexual reproduction. We'll talk next time about variation in the population.